Welcome to STEM Talk.
STEM Talk. STEM Talk.
STEM Talk.
STEM Talk.
Welcome to STEM Talk, where we introduce you to fascinating people who passionately inhabit the scientific and technical frontiers of our society.
Hi, I'm your host, Dawn Cornegas, and joining me to introduce today's podcast is the man behind the curtain, Dr. Ken Ford, IHMC's director and chairman of the double secret selection committee that selects all the guests who appear on STEM Talk.
Hello, Don. Great to be here. Our guest today is Dr. Matt Kaeberlein, a professor of pathology at the University of Washington, who investigates the basic mechanisms of aging to facilitate interventions that promote health span and lifespan.
So in today's interview, we talked to Matt about the biology of aging and what he's learned about slowing the aging process. And also some of Matt's most fascinating research has shed new light on rapamycin, the only known pharmacological agent to extend lifespan. His paper that reported short-term rapamycin treatment later in life could delay aging in humans received a lot of attention. And today we talked to him about that paper, as well as his research showing rapamycin could reduce the mortality of companion dogs. And that's research that landed on the front page of the New York Times and received prominent play in the national media. In addition to his work in his lab, Matt is also co-director of the Nathan Schock Center of Excellence in the Basic Biology of Aging and the founding director of the Healthy Aging and Longevity Research Institute at the University of Washington. And he's also the founder and co-director of the Dog Aging Project. Before we get to our interview with Matt, we have some housekeeping to take care of. First, we really appreciate all of you who have subscribed to STEM Talk, and we're especially appreciative of all the wonderful 5-star reviews. As always, the Double Secret Selection Committee has been continually and carefully reviewing iTunes, Google, Stitcher, and other podcast apps for the wittiest and most lavishly praise-filled reviews to read on STEM Talk. If you hear your review read on STEM Talk, just contact us at stemtalk@ihmc.us to claim your official STEM Talk t-shirt.
Today, our winning review was posted by someone who goes by the moniker Ski A6. The review is titled, Just Like School, Only Different. It reads, I should say only much better. It is wonderful to listen to people, Ken, Dawn, and guests, who can make a complex topic understandable and something you want to keep learning about. You'll use your brain, so watch out.
Well, thank you, Ski A6, and thanks to all of our other STEM Talk listeners who helped STEM Talk become such a great success.
Okay, and now on to our interview with Matt Kaeberlein.
STEM Talk. STEM Talk.
STEM Talk.
STEM Talk.
Hi, welcome to STEM Talk. I'm your host, Dawn Cornegas, and joining us today is Matt Kaeberlein. Matt, welcome to the podcast.
Thank you. It's a pleasure to be here.
And also joining us is Ken Ford.
Hello, Don, and hello, Matt.
So, Matt, let's get started. Where did you grow up?
So I was born in Erie, Pennsylvania, but my parents moved to Seattle when I was very young. So I grew up in the North Seattle area, actually not too far from the University of Washington, where I'm at now.
Is it true, a birdie told me, so I have this from a bird source, for what that's worth, that you spent a good deal of time in your youth up to no good? What did the bird mean by that?
Yeah, I mean, I think that's probably a fair assessment. You know, in some ways, I look back and I feel fortunate that I grew up a little bit before the age of the internet. So, you know, all of my activities weren't recorded for future public consumption. Yeah, I mean, I guess I would say that, so my parents got divorced when I was pretty young in about third grade. And, you know, I lived with my mom and my brother and she had to work full time. So I don't know that I was up to any less good than any typical, you know, child that age would be. You know, when your single parent is working a full-time job and you've got a lot of time on your hands. So, you know, but I think it's fair to say that I wasn't particularly interested in school. I wanted to have fun, spend a lot of time, you know, not doing homework and doing other things that looking back, I probably shouldn't have been doing. But I have to say, you know, it all worked out in the end. So I don't feel too bad about my misspent youth.
So, Matt, despite your home alone years, you always got decent grades, but it really wasn't until you went to college that you became studious, even though I understand, I think this is from a birdie as well, that you decided to skip college after high school. So what happened?
Yeah. So again, I think that's a pretty fair assessment. I mean, I feel very fortunate that school always came easy to me. So I never needed to do homework or study. I certainly didn't get straight A's in high school, but I got grades that were good enough to get by. And, coming right out of high school, again, looking back, I have to say I was extremely immature at that point. I did get accepted to the University of Washington and USC, but I honestly just wasn't ready to make the leap to college. So I spent about 3 years working at United Parcel Service, the morning shift. I started unloading trucks, and then, I was good enough at that they put me on loading trucks and sorting packages. And so that's really what I did for the first few years after high school. And I think, pretty quickly realized that's probably not what I wanted to do for the rest of my life. So I also started going to community college while I was working at United Parcel Service. And that's the point, I think, when I really started to take academics seriously. And I think it was just a matter of growing up and being ready to really, get interested and excited about learning.
Yeah. So leading to another question for you then, Matt, how did you end up at Bellingham at Western Washington?
Yeah, so I would say this has sort of been a pattern throughout my life, that a series of fortunate accidents sort of all came together and ended up in a really good place. So while I was working at UPS and attending community college, I met the woman who would go on to become my wife, and we started dating. And she was also completing her 2-year degree at the same time. And so, we decided, I think, pretty quickly that we were meant for each other, and started thinking about what we wanted to do with the rest of our lives. And so we started looking around for 4-year colleges to finish up our undergraduate degrees. And we took a road trip up to Bellingham, which is where Western Washington University is, and really just fell in love with the location and the campus. And fortunately, both got accepted into Western out of community college. And that's really how we ended up there. And again, I feel very fortunate that things worked out the way that they did.
Yeah. So now I have to ask you, as someone who didn't particularly like high school and spent 2 years after graduation working the morning shift at UPS, how did you decide to go into biochemistry?
Yeah, again, a really happy accident. So I certainly was not inclined towards biology when I started going to community college. So I was, again, one of these students who was very, so mathematics and quantitative stuff sort of came naturally to me. So, that's kind of where my interests were, as much as they were in anything academic at that point. But I had a really, really fantastic biology teacher at Highline Community College. And for the life of me, I wish I could remember his name, because he had a huge impact on me and really, I think taught biology in a way that resonated with me at that stage in my life, and really got me passionate about biology and evolution and natural selection and biochemistry a little bit. So that was really the moment that kind of shifted, my trajectory towards biology and biochemistry. And then when I was considering what to major in at Western, biochemistry is what I settled on. Again, honestly, at that point, not really knowing a lot about what biochemistry was beyond, the introductory biology class I'd had in community college.
So after that very positive experience at Western Washington, you travel across the country to Boston and to MIT for graduate school. MIT is good at many things. What in particular attracted you to select MIT among the other choices?
Right. So, again, I wish I could say it was maybe more informed than it was. The honest answer is, as I was finishing up my 4-year degree, so to take a step back, I majored in biochemistry when I went to Western. But while I was at Western, I also had a really great mathematics professor who invited me to attend class that he was teaching during the summer on formal proofs, and really talked me into getting a second degree. So I came out of Western with a biochemistry degree and a mathematics degree. And so my training at that point was really focused on, structural biology, biophysical chemistry. And so I was looking around at programs, and I applied at several places. I applied to both MIT biology and MIT chemistry and biomolecular structure and design here at the University of Washington. And I was actually leaning towards the biomolecular structure and design program at the University of Washington. The real reason why I ended up going to MIT is because my wife had identified a person that she wanted to work with for her PhD at Northeastern, which of course is in Boston. And, there was no way I was going to let her go. So of course I had to go to Boston. And that's the honest reason why I picked MIT. Why I picked the biology program was really a matter of, I think, culture, and it just felt like the right fit. When I went out, I went out to visit both chemistry and biology, and I just really liked the culture and the people that I interacted with in the biology program. And so that's why I ended up going to there.
We've had several STEM Talk guests who talk about following spouses, typically for graduate school, at least to the same city, so they could be together. At some point while you were at MIT, I'm not sure when this happened, you decided to focus your research on the biology of aging. How did that come about? What was there like an epiphany moment, or it just fell into place? How'd that happen?
Yeah, there really was an epiphany moment. So as I mentioned, when I went to MIT, I was very much thinking that I would work on structural biology, X-ray crystallography, something like that, something more quantitative, hardcore biochemistry or biophysical chemistry. And that really is because that's what my training was in as an undergraduate. At MIT at that point in the biology program, there's about a month between the fall semester and the spring semester where the undergraduates don't take classes. And in the PhD program in biology, they had all of the professors who were interested in taking graduate students give a seminar. So it's about a month where you have 2 or 3 seminars a day by different professors in the department. And it's really designed, I think, to expose the graduate students to all of the diversity of research happening in the department. And so it was during that time that I heard a seminar by Lenny Guarente, where he talked about how his lab was using genetics and molecular biology and biochemistry to study the biology of aging. And at that time, they were working in budding yeast, a single-celled organism. But it was just listening to Lenny give that seminar that really resonated with me. And I'm not exactly sure why, but I think it was because I'd never even considered that we could use genetics and molecular biology and biochemistry to study something as complicated as the biology of aging. And so I just really got turned on to that idea. And it obviously, fundamentally shifted my direction away from structural biology and biophysical chemistry into the biology of aging. And so again, I can look back at that moment when Lenny gave that talk and really see, how this had a really fundamental effect on the rest of my career.
So Matt, what happened after you finished your PhD?
So I started graduate school in 1997, and I joined Lenny's lab in 1998. And then I graduated in December of 2001. And I think at that point, I was maybe a little bit burned out on graduate school. I think that's not uncommon for graduate students by the time they reach the end of their PhD. So I looked at doing academic postdocs, had some actually really good offers when I look back now at the people whose labs I could have gone to. But instead, I decided to go into a startup biotech company that was spun out of George Church's lab by a former student of his named Pete Estep. And the company was called Longevinity. And it was really in the aging space where I had spent my graduate studies. And so I spent about a year and a half at Longevinity right out of graduate school. And again, I think, even though the company didn't end up, being successful, it was a really important and valuable experience for me just to see that side of the world and the way things operate at a startup biotech where you're coming in and there are only 3 people there and everybody does everything from the CEO all the way on down to, the most junior person at the company. So I have no regrets. And I think it was really a valuable learning experience.
Yeah, it's a really good experience. And then you ended up back at the University of Washington in the Department of Genome Sciences for your postdoc. And what kind of research did you do in the department? And then how did that relate to aging?
Right. So at the time that I was at Longevinity, my wife was finishing up her PhD at Northeastern. And then when she was very close to wrapping up, we sort of made the decision together that I would go back to academia and we would try to find postdocs at the same institution. And so we were looking between the Bay Area and Seattle. And, we were both very familiar with Seattle. Her parents had moved to Seattle from Fairbanks, Alaska by that point, and my parents were in Seattle. So it was sort of natural for us to really take a close look at the University of Washington. And I ended up joining Stan Field's lab in the Department of Genome Sciences at the University of Washington. And I went to Stan's lab for a couple of reasons. One is I really, really liked Stan's approach to science and the personality of the lab and the people that I interacted with. Looking back again, I have to feel so fortunate that I ended up with Stan as my postdoctoral mentor. He is just such a fantastic human being and a fantastic scientist. It had a huge impact on me. But his lab had also just recently taken an interest in aging, and they were working again in budding yeast, which was something that I knew very well from my graduate work. And so when I went to Stan's lab, I went there with the idea that I would do something related to aging in yeast, but really, without any specific project in mind. I also have to say that the University of Washington was a great fit for me at that point as well, because there was a very strong community of people there all working on different aspects of aging, not just in yeast, but all the way up through mice. So I was able to get a spot on the Genetic Approaches to Aging training grant. University of Washington had a Nathan Schock Center of Excellence in the basic biology of aging, which we still have. And so there was a great community outside of Stan's lab, all with shared common interests around the biology of aging.
It was during this time that I understand you decided to focus much of your work on answering the question, to what extent are the mechanisms of aging evolutionarily conserved? And that's a fascinating question, and you've become known for this. How did you arrive at this as your central question at the time?
So when I got to the University of Washington and started working in Stan's lab, I got together with Brian Kennedy, who was an assistant professor in biochemistry at the University of Washington at the time. And as it turns out, Brian was also a postdoc with Lenny Guarenti. We didn't overlap at all, but we kind of met each other once or twice. And Brian, as he had started his own lab at the University of Washington, had gotten back into studying aging. And so we sat down and had coffee and basically came up with the idea that much of the field at that point had gotten focused around a small number of genes. And one of the genes that a lot of people were interested in at that point was SIR2, which actually stemmed from my graduate work. And so we quickly realized that there was this whole area of unexplored territory with everybody in the field focusing on a small number of genes, and of course there are thousands of genes in the genome, even in yeast. Why don't we, kind of take a step back and try to figure out what else is out there? So this question of conserved longevity genes, actually, we really didn't start testing that until towards the end of my postdoc. We started by trying to take an unbiased approach to understand, other than the 3 or 4 things that everybody else was looking at, what else was out there. And so we started this unbiased screen for single gene deletions that extended lifespan. And it was a lot of work. I think people probably thought we were crazy to try to tackle that at that point. But as it turned out, we were fortunate that there was a lot of undiscovered stuff out there. And one of the things we found very early on was a gene called TOR, which has a homologue in mammals called mTOR, or target of rapamycin, which really set the stage for much of what I did in the rest of my career. But it was as we were going through this genome-wide deletion screen, and now we had, instead of 3 or 4 genes that were interesting, we had 40 or 50 genes that were interesting because we'd found new genes that affected lifespan. That's what really got me interested in this question of, to what extent are the genetic regulators of aging shared across different species? And really the driving force for that was my own frustration with the people who were working in mice and how dismissive they were towards the people who were working in yeast and C. elegans and fruit flies like myself. So I think at that point, there were really 2 camps in the field. One camp were the people working in invertebrate models, and the other camp were the people who'd been working in mammalian models for many years. And most of the people who worked in mammalian models were of the opinion that you couldn't learn anything about aging unless you were working in a mammalian model. So the mouse people thought that you could learn a lot about human aging by working in mice. The people studying aging in humans thought you could only learn about human aging by studying aging in humans. And those of us working in vertebrates, we didn't know, right? We were just trying to figure out the biology. So I got kind of frustrated with this idea that was, and you can go back and find papers where people wrote opinion papers that, yeast is a useless model for studying aging, or you can't learn anything about aging by studying C. elegans. I got kind of frustrated with that and said, well, that might be right, but nobody has actually tried to ask the question, can we quantitatively find evidence that aging is or is not evolutionarily conserved? So that was really the driving force that got me to really try to test that question. And then the way that we did that was to take all of the genes that at that point we had found could affect lifespan in yeast, and all of the genes that other people had found could affect lifespan in worms, and try to cross-validate by looking for orthologs that are shared between the 2 organisms. And it turned out that we could, in fact, find evidence that there is quite a significant overlap between genetic modifiers of lifespan in yeast and genetic modifiers of lifespan in worms. And that's pretty encouraging because the evolutionary distance between yeast and C. elegans is actually bigger than the evolutionary distance between C. elegans and humans. So it doesn't prove that those genes affect aging in humans. But I think at that point, it was really the first quantitative evidence that fundamentally, the genetic control of longevity does seem to be evolutionarily conserved.
So, the discovery by you and Mitch McVey that overexpression of SIR2 is sufficient to extend lifespan in yeast is credited with promoting SIR2 to the forefront of aging research. And SIR stands for silent information regulator, and SIR genes are determinants of lifespan in yeast mother cells. Before we talk about your paper, which appeared in 1999, can you give us and our listeners a quick primer on the SIR genes and their overall functions?
Sure. Right. So as you said, SIR stands for silent information regulator, and it was first studied in budding yeast. And really what that means is that the SIR proteins regulate gene expression. So silent information regulator, there are some parts of the yeast genome that are transcriptionally silenced. And so in the case of SIR2, that protein functions to inhibit the expression of genes around the telomeres and at what are called the silent mating loci in yeast, and then also affects transcription in the ribosomal DNA. So that's kind of what was known at the time I started my graduate work, was that there were these SIR genes, SIR2, 3, 4, that all functioned in a complex to regulate gene expression at those specific locations in the yeast genome. Nobody knew what their biochemical activity was at the time, but that was it was known that their function was to regulate gene expression.
So speaking of the 1999 paper that appeared in the journal Genes in Development, which is one of the top journals in that field, can you talk about this paper and how you demonstrated that increasing the dosage of SIR2 extended lifespan?
Sure. So as I just mentioned, at the time, we didn't really know much about what the biochemical function of SIR2 was. What we knew was that if you deleted SIR2 in a SIR2 mutant, you got hyper-recombination at the ribosomal DNA, and that led to a shortening of lifespan in yeast. And so when I started my graduate work, that was really my project, was to try to understand, what was the relationship between genome instability at the ribosomal DNA and lifespan. And so sometime in my second or third year in graduate school, Mitch and I came up with the idea that maybe one way to really test this would be to overexpress SIR2 and see what the effect was on lifespan. And actually, I think this is maybe a good time to talk about a conceptual point in aging that's actually really important. So I just mentioned that you could delete SIR2, so loss of function, and you got shortened lifespan. That is, I think, suggestive that gene may have a role in aging, but it's also important to recognize there are lots of ways to shorten lifespan that don't have anything to do with normal aging, right? I could spend my day walking across a busy street, not looking if cars were coming. My lifespan would be shortened. I didn't necessarily age at an accelerated rate. So the real definitive way to demonstrate that you've got a gene or a drug that affects the aging process is to increase lifespan. And so that was the rationale for doing that experiment. So we did that at the time by transgenically integrating a second copy of SIR2 into the yeast genome. We could verify that we increased the activity because we could see silencing at those loci. So again, remember, SIR2 decreases transcription. We had reporters where we could see that overexpressing SIR2 further inhibited transcription at those places. And then we measured lifespan and we saw that it extended lifespan. So that was, I think, really compelling evidence that SIR2, in yeast at least, was an important regulator of lifespan. You sort of started this by suggesting that that's what, really convinced people that sirtuins were important. I wish that was the case. I don't think a lot of people paid that much attention at the time. I mean, we published it in a good journal. It was an important paper, but I think what really got the rest of the field to pay attention was when Heidi Tissenbaum, who was a postdoc in the lab at the time, after our work, went and tested whether the C. elegans homologue of SIR2, SIR2.1, could also affect aging in C. elegans. And it was when she showed that it wasn't only a yeast thing, but this seemed to work in a multicellular animal, that I think that's when people really started to get interested in sirtuins.
So after you arrived at the University of Washington, you started looking at two simple organisms, so yeast and C. elegans, to see if there were key aspects of aging that could be related to humans. And then in 2008, you and your colleagues were able to publish a very important paper in the journal Genome Research that demonstrated quantitative evidence for conserved longevity pathways between different eukaryotic species. So can you talk about how, partly because of this paper, almost everyone now agrees that there are key aspects of aging that are shared across all animals, even down to yeast? It's pretty cool.
Right. Yeah, thanks. So again, I think at that time, we knew that there were a few genes that seemed to play an important role in the aging process in these different model organisms that people were studying. So, back then, and this is still true today, that the common model organisms that people use to study aging and many other biological processes are budding yeast, C. elegans, fruit flies, and mice. And so we knew that there were some genes that affected aging in all of those organisms. And it looked like there were a few pathways that might be important across those species. But, as I was mentioning earlier, particularly the people who studied aging in mammals were still pretty dismissive of invertebrate model organisms at that point. And there was this perception that you couldn't really learn a lot that would be important for human aging by studying aging in these very simple animal models or cellular models. And so we set out really to try to test that, because I think you can make arguments both ways, but until you actually do the experiment, you're really not going to know. And so we decided to test whether or not we could find evidence for conservation of genetic control of lifespan between yeast and C. elegans. And one important thing to mention about this is this was a really nice comparison, both because of the evolutionary distance between yeast and C. elegans, and also because yeast, the way we were measuring lifespan in yeast, is primarily a mitotic model of lifespan. And what I mean by that is we were measuring replicative lifespan in yeast, which is defined by how many times can a mother cell divide before it goes into senescence. So it's a mitotic cell division assessment of aging. In C. elegans, when you're talking about lifespan, you're talking about adult lifespan. And C. elegans, with the exception of the germline, are completely post-mitotic animals. So these were sort of potentially fundamentally different aspects of biological aging. And so if we found evidence for conservation, we thought that would be quite compelling, that those pathways seem to be fundamentally involved in the aging process, and potentially a reason to go look at those pathways in mammals. And so the experiment we did was conceptually very simple. In reality, quite tedious and time-consuming, was to just look at all of the genes that we knew affected lifespan in yeast and all of the genes that we knew affected lifespan in C. elegans, do some mapping to identify the homologs and orthologs between the two organisms, and then test all of those homologs and orthologs that hadn't previously been tested. And at the end of the day, what we were able to show is a significant enrichment in genes that affected lifespan positively between the two organisms. And again, this gets back to that conceptual point that I was making a moment ago. We were really limiting ourselves to cases where we could show lifespan extension, because shortening of lifespan is a non-specific phenotype, but lifespan extension is very specific. And so we were able to show that we could identify a significant enrichment when looking among orthologs for lifespan extension across the two species. And so that was really the first quantitative evidence that genetic control of lifespan is shared across evolutionarily divergent species.
So, Matt, this is probably a good time to talk about your lab and the work that you're doing at University of Washington. So, one of the mottos that's on your lab's website is "Live long or die trying," which I love, by the way. But in addition, you like that too, Ken?
Yeah, it's cool.
But in addition to the Kaeberlein Lab, you also are the director of the Healthy Aging and Longevity Research Institute and the director of the Nathan Schock Center of Excellence in the Basic Biology of Aging, as well as the director of the Biological Mechanisms of Healthy Aging Training Program. So for someone who says he had a misspent youth, you certainly seem to be making up for that. So first, can you tell us a bit about your lab and how its work intersects with all these other programs?
Sure. So, I mean, I'm tired now listening to all the things that I have to do.
All that stuff.
So, I mean, I think, the way I've operated my lab from the very beginning is to focus what we work on, to some extent on the biology of aging. But within that, really give ourselves flexibility to go after interesting things that we uncover. So I've intentionally tried not to limit what we will study beyond everything that we study in the lab should be relevant for the basic biology of aging. And, I mean, people will have different opinions on that approach. I think some of my colleagues might call me unfocused, but I think it's been, first of all, it's fun. That's the way I like to do science. And I think we've been successful at making contributions to the field by taking that strategy. But the way it sort of intersects with those other titles that I have is really all of those are about building and maintaining a community beyond my lab at the University of Washington with a shared interest in the biology of aging. So the Nathan Schock Center is an NIH-funded center of excellence in the biology of aging. The training grant, the Biological Mechanisms of Healthy Aging training program is a training grant that funds 6 graduate students and 6 postdocs, not in my lab, but throughout the university around the biology of aging. And HALO, the Healthy Aging and Longevity Research Institute, is, an umbrella organization in some ways that can support initiatives at the University of Washington related to the biology of aging. So, I think everything's more fun if you've got a community of like-minded people who share your interests and your mission. And so I think that's been really part of the motivation there, is to help build and maintain that community at the University of Washington.
Yeah, I like the way you set your lab up, and you described it has a general focus, but within that general focus, there's room to move and to explore new direction.
So, Matt, I want to back up and talk about a couple of papers you had back in 2004 and 2005. So calorie restriction has been shown to increase lifespan in yeast, worms, flies, and in mammals. And the molecular mechanisms for this phenomenon, however, has been pretty unclear. In 2004, you and Brian had another paper that made a significant impact in the caloric restriction world. And it disproved the model that caloric restriction acts solely through SIR2 to mediate lifespan extension.
Then you followed up with another important paper in 2005 that specifically identified mTOR as an aging gene in the same pathway as caloric restriction. Can you talk a little bit about the origins of these two studies and their significance?
Sure. So I'll take a step back and talk a little bit about how this idea that caloric restriction was acting through SIR2 came about, because I think this whole sort of story has some important lessons beyond, this specific pathway for the way that we think about and approach models in scientific research. So this idea that caloric restriction activates SIR2, and that's how caloric restriction extends lifespan, really came out of my graduate lab, the Garenti lab. And some of that work was done while I was still a graduate student there. So at the time that this was being studied, what we knew was that caloric restriction was really the gold standard for affecting lifespan in different animals. And in fact, caloric restriction was first described to extend lifespan in rats in 1935. So it has been studied extensively in mammals for many, many decades. And while I was a graduate student in the lab, Suju Lin, who was a postdoc at the time, showed that you could increase lifespan in budding yeast by caloric restriction, specifically by restricting the amount of glucose in the media. And so she was doing that work right around the time that we published that overexpressing SIR2 could extend lifespan. And so she did a series of experiments in a particular yeast strain. And what she showed was that if you deleted SIR2, you no longer got the lifespan extension from caloric restriction. And that really was the primary result that led to the model that caloric restriction activates SIR2 and increases lifespan.
You can certainly see how that would lead people to think that at first blush.
Right. And it's consistent with the model. I mean, I think that's where we have to always be thinking is, and that's the goal, right? The goal should be to try to disprove your hypotheses. And so certainly that's consistent with the model, but there are also other alternative explanations that could explain that result. And as I mentioned previously, SIR2 mutants are short-lived, right? So to go back to my example of if you're constantly walking across a busy street and not looking for cars, there's a good chance you'll shorten your lifespan. Caloric restriction isn't going to extend your lifespan if you get hit by a bus, right? So it may or may not have anything to do with whether or not those are mechanistically acting, by similar mechanisms in the same pathway. So I think that's the conceptual underlying point here that's important. So, but it was consistent with that. And that model kind of got to be, somewhat dogma in the field. And so when I went and started my postdoc at the University of Washington, Brian and I, were both a little bit dissatisfied with the data around that model. Not that we thought it was wrong, but we knew that there were some experiments that hadn't been done yet at that time. And one of those experiments was to combine caloric restriction with SIR2 overexpression, because both of those alone extend lifespan. And so the question is, what happens if you do them together? That seems like an obvious experiment, right? You would think that experiment would have been done already. The reason it hadn't been done is because the genetic backgrounds where I had shown that SIR2 overexpression extended lifespan was different than the genetic background where Suju had shown that caloric restriction extended lifespan. And what never got in the literature up to that point was that those two interventions didn't work in the other genetic background, right? So in other words, the genetic background where SIR2 overexpression extends lifespan, caloric restriction didn't, and vice versa. That wasn't in the literature. So I think the example here is that sometimes things that are missing from the literature can be really important. And if you don't have that knowledge, there's no way to understand, weaknesses in the model. So Brian and I had that knowledge because we were in the lab when the work was being done. And so we were able to understand that what we needed to do was to find a genetic background where both interventions worked individually, and then we could combine them and test the model. And so that's what we did. So we went and we worked in a third genetic background, which also happened to be the genetic background where the yeast deletion collection had already been made. Because remember, this isn't happening in isolation, right? We were planning for this large scale genetic screen, this unbiased genetic screen, to look across the entire genome and find out what affected lifespan. So of course, we wanted to work in the strain background where that library had already been made. But we were fortunate that strain background also responded to caloric restriction and also responded to SIR2 overexpression. So we could put them together. And what we saw was that it was an additive effect on lifespan, which is not, it doesn't prove that they're in different pathways, but it's suggestive, right? That you're hitting two different pathways, and then when you combine them, you get double the effect, right? So that was our first hint that maybe that model was imperfect. And so we went forward and did a series of additional experiments where we could actually create a genetic background lacking all sirtuins in yeast and still see lifespan extension from caloric restriction. And so I think that is pretty definitive that you don't need sirtuins to get lifespan extension from caloric restriction. It doesn't prove that none of the effects of caloric restriction are due to sirtuins, but it certainly says that they're not necessary to get lifespan extension from caloric restriction. I think what's interesting about this is that even still, if you ask people in the field, you will still find people who very much believe that caloric restriction acts only through sirtuins, even in mammals, and that's the mechanism. And so the reason I bring that up is it is very, very difficult, and it takes a very, very long time, no matter how strong the data is, to overturn dogma once it becomes dogma in science. It takes decades. And so we're still fighting that battle a little bit.
That was a great explanation. We've had several guests at STEM Talk who discussed TOR, in particular mTOR, in some detail. And I wondered if you would dive a little bit into your TOR-related work.
Sure. Yeah. So we really got interested in TOR by accident. I wish I could say that, I was really smart and I knew that would be important and went and tested it, but that's not the case. So, about the time we were finishing up these experiments looking at SIR2 and caloric restriction, at that point we knew that there was more to caloric restriction than SIR2, but we didn't know what. We had also started this unbiased screen of the yeast deletion collection. And what we were doing there, again, very conceptually simple. The collection was arrayed in 96 well plates. Each well contained a yeast strain lacking one non-essential gene. And there were about, I think, 4,800 strains. And so we just started one day with, randomly picking a plate out of the freezer and testing one by one each of these strains for their effects on replicative lifespan in yeast. And we were really pretty lucky that in the first 5 plates that we chose, the TOR1 gene deletion was one of the strains that was in that plate. So we found out early on in this screen that deleting TOR1 in yeast could extend lifespan. And so I didn't know much about TOR at that point, and I went and did a literature search at the time, and pretty quickly found out that TOR in mammals, or mTOR, which stands for mechanistic target of rapamycin, was involved in sensing nutrients and regulating growth and development. And so that connection to nutrients pretty quickly made me think that maybe this had something to do with the lifespan extension from caloric restriction. And so we did a series of experiments that were published in 2005 in a paper in Science, where we made a case that caloric restriction in yeast was extending lifespan by inhibiting TOR. And that was, I think, again, we were really lucky in the timing. And this is another sort of interesting thing about biomedical research is sometimes, because of a twist of fate, multiple labs all end up converging on a similar discovery completely independently. So right around the same time that we accidentally found TOR deletion extending lifespan in yeast, other labs were finding in C. elegans and in fruit flies that inhibition of TOR could also extend lifespan. And so there were a series of 3 or 4 papers published all within a year of each other showing that inhibition of TOR could have an effect on lifespan in yeast and C. elegans and fruit flies. And then, of course, based on our paper, that this seemed to be part of the mechanism downstream of caloric restriction for extending lifespan. And that's really how TOR got its start in the aging field. I think up to that point, people had really been thinking about TOR as a regulator of cell division. People were very interested in inhibiting TOR for anti-cancer effects. And of course, by that point, the drug Rapamycin, which is a specific inhibitor of mTOR, had already been FDA approved for organ transplant use. So there was already a drug in use in people that's a specific inhibitor of TOR. And of course, it was that link between Rapamycin and TOR that led us to go on. And in 2006, we published a paper showing that Rapamycin treatment could increase lifespan in yeast. And that was really the first paper showing that Rapamycin could increase lifespan in any organism.
And your lab then followed up on these 2 papers with a study that showed fasting extends lifespan in worms more than caloric restriction, although in a kind of a weird kind of context. So these worms were fasted for their entire and comparatively speaking, brief adulthood. And the paper that came out from this study was published in Aging Cell under the headline Lifespan Extension in C. elegans by Complete Removal of Food. So, in other words, starvation maximizes adult lifespan in these particular worms. Can you tell us a little bit about this very intriguing study?
Sure. So again, I think this is kind of a good example where you have to be careful in science not to go into your experiments with preconceived notions, right? About what's going to work and what's not going to work. So the reason we did this experiment was really to try to develop a reproducible model of caloric restriction in worms by reducing the amount of food that the worms got. And in the lab, C. elegans eat E. coli. So the bacterial food is what they eat. And so I would have never predicted before starting this experiment that taking all of the food away would give the biggest effect on lifespan. What we really did was a dose response. And what we expected to see was an increase in lifespan as we reduced the food to some point. And then starvation would set in and the worms would be short-lived. What we found was that we could keep reducing the food till there was nothing, and that's when they lived the longest. Now, I do have to say, you have to do this experiment in sort of a couple of special ways. Otherwise, you won't get that result. One is you have to start the caloric restriction when the worms are adults, right? So if you do this while they're still developing, if you totally take all the food away, they won't finish development, which makes sense. And you also have to inhibit reproduction. And we did this. We inhibited reproduction in worms as part of our standard protocol, which is why we got this result. If you don't inhibit reproduction and you take all the food away, the worms will be short-lived. So it's a little bit context dependent where you get this maximized lifespan upon complete removal of food. But if you wait till adulthood and you inhibit reproduction, then taking all the food away gives you the biggest lifespan extension. Now, of course, that's going to be not the case in mammals and not the case in people. We know that complete food removal eventually will limit your lifespan. But at least in the context of nematodes, that's where you get the biggest effect. And I think, you can make some interesting evolutionary speculation about why that might be. And what we and others have followed up on this is more, trying to understand the downstream mechanisms for how bacterial deprivation, that's what we call the complete food removal, for how bacterial deprivation extends lifespan in worms.
This is certainly one experiment that you hope not to see replicated in humans. IRB would have a fit.
Right, yeah.
It would never get passed.
Well,
actually, though, that raises an interesting point because in mice, the largest lifespan extension from caloric restriction is at about 65% reduction in calories. So at that level of caloric restriction, you get about a 65% increase in lifespan. And those experiments were done in the 80s and maybe early 90s. I think it would be very difficult for anybody to do a caloric restriction experiment at that level of restriction today for exactly the reasons that you were just alluding to on the human side. It would be very difficult to get an IACUC, an animal use committee, to approve a 65-70% restriction of calories in mice. So, sometimes the experiments that you can do in science are limited by obviously what is ethically acceptable at that time. And obviously what's ethically acceptable changes with time as society changes.
Sure. A few years after this research we just discussed, you started looking into the drug rapamycin further. And in particular, you were looking at the role it plays in increasing the lifespan of rodents and how it delays age-related dysfunction. Can you walk us through this 2016 study, which is pretty significant because it suggests that even short-term rapamycin treatment and even late in life can not only delay aging but also influence things like cancer prevalence and perhaps even modulate the microbiome?
So let me just take a minute to walk through the history of rapamycin, especially with studies in mice. So I mentioned previously that we did a study that was published in 2006 where we showed that you could treat yeast cells with rapamycin and extend lifespan. About 3 years later, a paper was published by the NIA Interventions Testing Program showing that you could start treating mice at 20 months of age with rapamycin and increase lifespan in mice. And so that study was, I think, foundational for the field in a couple of ways. This was really the first time that anybody had ever shown that you could start a treatment in mice in middle age and get really a pretty robust effect on lifespan. So they started the treatment at 20 months, which is about the mouse equivalent of a 60-year-old person. And when you think about it from the perspective of translation, potentially someday doing this in people, that's really important because you want an intervention that you can start in middle age when people start to see the declines that go along with aging. You don't want something that teenagers have to start taking for the rest of their lives. So that was already known at the time that we started our 2016 study, that you could start treatment at 20 months of age. And if you continued it for the rest of life, that you could get pretty robust lifespan extension. It was also just starting to emerge that treatment with rapamycin in middle age for 8 to 12 weeks could restore some aspects of age-related function. So there had been a couple of papers published showing that 8 weeks of rapamycin treatment could improve heart function in old mice. And then a paper from Pan Zheng's lab, which is, I still think, really underappreciated, had shown that 6 weeks of treatment with rapamycin in old mice could rejuvenate the immune system and actually allow the mouse immune system to respond much, much better to a flu vaccine. So we kind of knew that when we started this study. This was around probably 2013, 2014 when we started this. And so the question we really wanted to ask was, could we do a short-term treatment with rapamycin in old mice, and what benefits would we see? And so the design here is very simple. We took 20-month-old mice. We tested a couple of different doses of rapamycin. They either got the control or rapamycin for 12 weeks, and then we stopped the treatment. And we didn't do anything to them at that point except let them live out the rest of their natural lives. And what we observed was we got pretty robust lifespan extension in the mice that got rapamycin. We also saw that their microbiome was remodeled in interesting ways. We also saw evidence that they were more active, had a little bit better strength and mobility. And we saw changes in the types of cancer that the mice got. So, that was, I think, suggestive that you get long-term persistent effects on aging from a short-term treatment with rapamycin during middle age.
STEM Talk is an educational service of the Florida Institute for Human and Machine Cognition, a not-for-profit research organization investigating a broad range of topics aimed at understanding and extending human cognition, locomotion, performance, and resilience.
So this seems like a very significant finding for humans, and it deservedly garnered attention. But it wasn't until you started treating dogs with rapamycin that your research ended up on the front page of the New York Times. Before we start talking about this research, I'm curious how you became interested in studying dogs and aging in the first place. We're just kind of guessing there's a good story there.
Yeah. So again, as we've talked about, I've been studying aging since I was a graduate student. In the late 1990s is when I started. And I've also always been a dog person. I've had dogs my entire life growing up, and my wife and I have had dogs the entire time that we've been together. In fact, our first child was our first dog together. So we had a canine child before our 2 human children. And I've always loved animals. But it had never really occurred to me that it might be possible to study the biology of aging in companion animals and in pet dogs in particular, until a series of conversations with my colleague, Daniel Promislow, who's also at the University of Washington, where, he and Kate Creevy, who's at Texas A&M, had really started thinking about, could companion dogs be used as a model to understand the biology of aging? And so I had a series of conversations with Daniel about this, and I thought it was great. I mean, I loved the idea of studying aging in pet dogs. But what really turned me on to the idea of the Dog Aging Project was actually doing something about aging in pet dogs. And to be completely honest, I want my dogs to live longer, healthier lives. And so that it was when I had that sort of, eureka moment where I'm like, wow, we really might be able to make pet dogs live longer, healthier lives, that, I sort of, at that point, I knew I had to do it. And so I started, this was probably around 2014 or 2013 when this happened. And so I started really trying to think about, what would it take to actually test whether or not something like rapamycin could increase lifespan and improve health span in companion animals. And so it's been a long process to get here, but we're actually at the point now where we are testing whether rapamycin can increase lifespan and health span in pet dogs.
Is your dog one of the subjects?
No, and the reason for that is really because of a conflict of interest, right?
So,
we recognized early on that none of the dogs for the Dog Aging Project team really could be involved directly in the clinical trial because of the appearance of a conflict of interest. Whether my dog is on rapamycin or not, I'll let you decide what you think.
Yeah, I have we've had several guests on the podcast that when I asked them that question with respect to themselves, they gave me a similar answer.
Yeah. I mean, I think it's important, right? Again, our intention all the way through has been to try to do this clinical trial at the very highest standards, because it's really important, right? Regardless of what result that we get, it's really important that people can have confidence in the outcome. And so, we have been very careful at every stage to really try to make sure that we're doing this at the highest standards, the same way you would do a human clinical trial.
No, that's great. And we're glad that you are. Earlier this year, you had an article in Nature where you explained that the primary goal of the Dog Aging Project, and it's so interesting, is to generate and facilitate discoveries on the fundamental biology of aging. And you just discussed this. However, in that article, you pointed out that the impact of the Dog Aging Project will go far beyond this goal. Could you elaborate a little bit about what you meant by that?
Sure. So the Dog Aging Project overall really has two goals, I would say. One is to understand the biology of aging, and that's in companion dogs, and that's through a large-scale longitudinal study of aging. And then there's the clinical trial, which I mentioned previously, where we're really trying to test whether rapamycin can affect aging in dogs. But within, or maybe overarching to those two goals, is to really try to stimulate scientific discovery as broadly as possible. And so the Dog Aging Project is an open science project. All of the data that we collect, particularly from the longitudinal study, is being made available to the scientific community. And I think that there will be many applications of that data that other scientists will be able to take beyond what we're going to have the bandwidth or maybe even the expertise or interest to study. So, as an example, I think one of the things that the Dog Aging Project will contribute is really a better understanding of what normal aging looks like in companion dogs. And it's sort of surprising because that understanding doesn't exist, at least in any formalized way right now, because geriatrics has never been a specialty in veterinary medicine. So we found pretty quickly, when we were trying to design this clinical trial, there really isn't a good source for looking at, what frequency of dogs as they age show age-related declines in heart function or age-related valvular regurgitation in the heart. So I think we're going to find all sorts of interesting information about what the normal aging process looks like in dogs that will be of use to veterinarians that are trying to treat companion animals. Likewise, we're going to be collecting microbiome data from 1,000 dogs every year. And our goal is really to try to understand correlations between the microbiome and health outcomes during aging. But of course, the microbiome is important for all sorts of other disease processes that people who have expertise in the microbiome might want to look at, or the relationship between microbiome in dogs and microbiome in humans. So I think there are a variety of applications outside of the biology of aging where the data that we collect will be valuable for the broader scientific community.
Yeah, it's really interesting research. And we're going to go back to rapamycin in humans. So recently you had a paper in eLife that looked at whether rapamycin could rejuvenate oral health in mice. So periodontal disease is something that particularly affects the elderly, and currently there's no therapy that can effectively reverse periodontal disease. But this study had some promising results, didn't it? And can you talk about those a little bit?
Right. So this has been a really fun project for me. And again, I wish I could take credit for it, but this was not my idea. So the whole reason we started looking at periodontal disease was because a dental PhD, so DDS PhD student, came and talked to me one day. And he made the point, which I had never thought of before, that periodontal disease is a disease of aging, just like most cancers, heart disease, kidney disease. And if you look at the increase in risk, it goes up dramatically with age in people. And as you just said, something like 2/3 of elderly adults suffer from periodontal disease. And if you have periodontal disease, you're at higher risk of dementia and diabetes and cardiovascular disease. So there's some link between having periodontal disease and other diseases of aging. And so John On was the graduate student who really came up with this line of research. And I, when he told me that, it sort of clicked all of a sudden that, well, maybe the same pathways that affect the biology of aging would have an impact on periodontal disease. And so we designed a way to try to test that in mice. And so the first thing that John did was showed that as mice age, normal aging in mice, they do develop all of the clinical signs of periodontal disease in humans. And so that's inflammation of the gums, pathogenic remodeling of the microbiome, and loss of bone around the teeth. So normal mice, normal aging show all of these changes. And so then we asked what would happen if we treated those mice with rapamycin for 8 weeks. And what we saw was a reversal in all 3 of those clinically defining features of periodontal disease. So I think that's pretty exciting. And I think, we don't know yet whether that's going to work the same way in people, but I like this because periodontal disease is an endpoint that is nice from the perspective of a clinical trial, because you have very quantitative things that you can look at. You can look at inflammation of the gums, and dentists are very adept at doing that. You can look at bone around the teeth by X-ray, and you can look at the oral microbiome. And so John now is in the process of designing and executing a clinical trial at the University of Washington to test whether or not rapamycin can reverse periodontal disease in elderly people.
It's really interesting work. And so we're going to change topics now and talk about another recent paper that came out of your lab. And this was a review summary that appeared in the journal Science last year that looked at anti-aging diets. So bookstore shelves are full of anti-aging titles these days, and you set out with this review to separate fact from fiction. As we discussed earlier, caloric restriction is the oldest and most studied intervention that has been shown to increase lifespan. And in this article, you evaluated several of the most popular anti-aging diets and compared them with classical caloric restriction. So can you give us and our listeners just an overview of the interventions that you evaluated and what insights that you gained?
Sure. So we looked at a variety of nutritional strategies that people have been studying in the context of the biology of aging and really tried to ask the question, how do those compare to true caloric restriction? And so just for definitional purposes, true caloric restriction is really restricting all of the macronutrients, so carbohydrates, protein, fat, basically the diet, by a fixed amount and supplementing with micronutrients and vitamins. So I think, maybe the more precise term is caloric restriction with appropriate nutrition. So it's really just restricting the diet across the board. And then we compared that to studies of different nutritional strategies. And there are several, as you mentioned, that have sort of been popularized in the popular media and in diet books. So among those are the ketogenic diet, protein restriction, fasting, intermittent fasting, time-restricted feeding, and a low-fat diet, or, the sort of Mediterranean diet, although that really hasn't been studied extensively in mice. There isn't really a good model for the Mediterranean diet. And so what we did was we really tried to take a close look at the literature and say, if you compare the studies that report lifespan extension or health span extension from these different nutritional strategies in mice, mostly, we started in mice, how good are the data? And I think what we found is that, there's evidence for health benefits from all of these different nutritional strategies, but none of them are anywhere near as effective as true caloric restriction. And one of the real confounding factors pretty much across the board is that the majority of studies that have looked at these different nutritional strategies typically don't control for caloric consumption. And so what I mean by that is a lot of the studies where people will claim that intermittent fasting, for example, extends lifespan and slows aging in mice, when you look at how much the mice actually ate, they're calorically restricted. So when you see these benefits, you don't know if it's the intermittent fasting or if it's the caloric restriction that led to the health outcomes. So I think that's kind of the take-home message, really, is caloric restriction works. It works pretty robustly. And none of these other interventions work nearly as well, at least for lifespan, in mice as true caloric restriction does. There's a lot of other nuance in there that we can dive into, but I think that's probably the most important thing that we found. I think the other thing that's important to note is that when you look at caloric restriction, again, at least in mice, there's a very strong genetic component to how individuals respond to caloric restriction. And this is also true, it turns out, in fruit flies and in C. elegans and in yeast. If you look across many different genetic backgrounds, what you find is that about 2/3 of the genetic backgrounds show a lifespan extension from caloric restriction, but about 1/3 actually have their lifespan shortened. And so, I think that should suggest some caution when we start making recommendations for humans, because obviously humans are genetically diverse, right? And we really don't have a good understanding, even in laboratory animals, what the mechanisms are there or how to predict which genotypes will show a positive response and which genotypes will show a negative response. And we really have no idea in humans. So I think we just wanted to raise awareness that there is going to be an individual component to how people respond to caloric restriction and almost certainly how people respond to these other nutritional strategies.
Also, that literature, the nutrition literature, is notoriously shabby in my view. And so if you look at the diets and they're all over the place, even with the same label, so you'd say ketogenic, right? Well, then you look at what they actually fed and they range from, they have a wide disparity in what falls under that title.
Yeah, I think that's fair. I think, doing nutritional epidemiology in humans is really hard. So I'm not disagreeing with you that the studies have a lot of weaknesses, but I also think we have to recognize it's just really, really hard to do those studies in a clean way. In mice, though, you're absolutely right that it's kind of all over the place in what the exact composition of the food is. And as I mentioned, I think the other, big confounding factor is very rarely do people carefully control for caloric consumption. And so again, you don't know if the effects you see are due to caloric restriction or due to the diet and the composition of the diet.
And the genetic differences you mentioned in the mice themselves.
Yeah.
I agree about the difficulty of doing nutritional epidemiology in humans. Given the difficulty, a degree of modesty about the claims would seem appropriate.
Yeah, I think you said it better than I could.
So, Matt, we've done some studies here at IHMC, as you may or may not know, about ketone esters, mostly looking not at disease or lifespan, but on performance. But you mentioned in your article that there are studies and findings that are highly suggestive that ketone esters by themselves might have anti-aging properties, and there's an emerging literature related to that. Could you just give us a quick update on that from your perspective?
Right. So what we can say for sure is that a ketogenic diet in mice can extend lifespan. But again, the effects are not as robust as lifespan extension from caloric restriction. I think the other thing that's useful to note about ketogenic diet studies is what you have to do to induce an animal to go into ketogenesis is very different in different organisms, right? So in mice, the ketogenic diets are extremely low in carbohydrates, much lower than you even would go in humans in order to induce significant ketogenesis. And, we've thought about this a little bit in dogs, and it turns out in dogs, you have to fast a dog for a couple of days, maybe even 3 days to get them to go into ketogenesis. So just what it takes to actually induce nutritional ketogenesis is very different. So then the question is, of the effects that you see from a ketogenic diet, how much of those effects are due to the production of ketone bodies themselves and the metabolism of ketone bodies themselves versus other aspects of the diet, right? Like weight loss, for example. And so the question is, would it be sufficient to just treat with ketone esters to induce some of these beneficial responses that have been associated with a ketogenic diet? So I know there are lots of people studying this right now in the aging field.
Definitely.
I haven't seen a lot of literature yet that you can get robust effects on lifespan and health span from supplementation with ketone esters. I know there's a lot of unpublished data that hasn't come out yet. And so I think we're sort of waiting to see. Again, I think the important thing will be when that data gets published to really take a close look and evaluate how big is the lifespan effect and what might the other confounding variables be. I have no doubt that ketone esters have potent biological effects, right? But I think we kind of have to wait for the data to be published to really evaluate how robust simply supplementing with ketone esters will be compared to, a dietary nutritional strategy that induces ketogenesis.
And in the case of humans, discovering strong effects on longevity will be hard and long to come by. But you can see the effect it has on the various pathways, some of which you've identified that seem related to longevity. Earlier, you talked about the difference in animals and the time it takes for various animals to come into a state of ketosis when subjected to starvation. This, as you probably know, is thought to be connected to the energy requirement of that animal's brain, and how quickly the animal would be in crisis without an alternative energy source. And that kind of makes sense, right? If you look at humans, dogs, it seems to go right in order, right? Humans, dogs, mice. So I thought that was a nice point you made. Toward the end of that same article we're discussing, you commented that a case can be made that mTOR complex 1, and we've talked about this earlier today, is a particularly relevant and robust molecular transducer of diet-induced anti-aging signals. And although we did talk about it, I think this might be a good time to loop back to that, because that's really one of the key points when we talk about mTOR and diet together. I think that's something that you could maybe touch on one more time.
Sure. Yeah. And I mean, I think this really stems from the question. We know these different nutritional strategies that are, all somehow related to caloric restriction can all have effects on lifespan and health span, at least in certain contexts. And, the question is that because there's a shared downstream mechanism? And it turns out you can find, very good evidence in the literature that with all of these nutritional strategies that affect lifespan, that they can also inhibit mTOR. Now, that doesn't prove that they're extending lifespan by inhibiting mTOR, but we already know that inhibition of mTOR is sufficient to extend lifespan in a fully fed state. And so it certainly is suggestive, I would say, that at least part of the mechanism of caloric restriction and several, maybe all of these other nutritional strategies is through inhibition of mTOR. But I also think it's really important to note that inhibition of mTOR in the context of caloric restriction biologically is going to be somewhat, maybe quite different than inhibition of mTOR in the context of treatment with rapamycin. So caloric restriction, and people get upset when I say this, but I'm going to say it anyways because it's true. If caloric restriction were a drug, it's just about the dirtiest drug you can imagine. And what I mean by that, so when people talk about a dirty drug, what they mean is it doesn't have a single biochemical target, right? Caloric restriction probably hits hundreds of different biological targets in an animal, whereas rapamycin is extremely specific. It hits pretty much one, and that one target is mTOR complex 1. So the biological output of caloric restriction and rapamycin treatment are going to be overlapping, but they're also going to be very distinct, right? So I think it's important to keep that in mind when we think about mechanisms. And so caloric restriction is going to do a lot more biologically than rapamycin treatment will. And that could be good, bad, or indifferent, right? But I think it's important to just keep in mind the complexity that you're dealing with when you're talking about something like caloric restriction. And then when you think about these other nutritional strategies, protein restriction, for example, that's still going to be pretty dirty, but it's probably more specific than caloric restriction in terms of the number of biological targets that get hit. But again, all of these nutritional strategies share at least transient inhibition of mTOR and mTOR complex 1. And so we simply just, proposed the model that maybe inhibition of mTOR complex 1 is a common downstream mechanism that links these different nutritional strategies and contributes to the effects on lifespan and health span. And then I think, the other, obviously, another interesting question is what else might contribute. And certainly there's evidence for other things like FGF21 potentially playing a role. And then what's downstream of mTOR that's important for lifespan extension. And that's really where, a lot of my attention these days is going is trying to understand how is inhibition of mTOR affecting lifespan and health span, and then also thinking about what does that mean for the kinds of changes we might want to look at following rapamycin treatment in both dogs and in people.
Quick question, a side question. Do you know which complex? Is it complex 1, mTOR complex 1, that's involved in muscle protein synthesis?
So this is really interesting, right? Because mTOR acts in two complexes, mTOR complex 1 and mTOR complex 2. That's true yeast all the way through to humans. mTOR complex 1 inhibition is, I think, where the most evidence supports effects on lifespan and health span. And rapamycin is a specific inhibitor of mTOR complex 1. And mTOR complex 1 is the primary complex that promotes protein synthesis, which has been what people have studied in the muscle field, right? So if you inhibit mTOR complex 1 enough, you will impair synthesis of new muscle.
That's what that's what I understood. This was off the record. I was just wanting to learn this because I had thought that was the case, and it would be great if it were otherwise, because then you could use rapamycin to hit mTOR complex 1 and not have the undesirable effects.
Yeah, I actually think this is a pretty important point. So it doesn't have to be off the record, because you get this question asked a lot. So I think 10 years ago, 15 years ago, if you were to ask a muscle biologist about rapamycin, they would say that rapamycin will cause a loss of muscle mass and that it would accelerate sarcopenia, which is the loss of muscle mass with aging, because mTOR complex 1 is required for synthesis of new muscle. So that's what they predicted. It turns out they were completely wrong. So if you treat a mouse or a rat with rapamycin, it lives longer and it has preserved muscle mass with aging, and it impairs sarcopenia. So I think it's really, really important to recognize that just because mTOR complex 1 is required for a certain process, partial inhibition of mTOR complex 1 with rapamycin in a biological context is not necessarily going to impair that process. The same thing was true with cognitive function. So mTOR is required for formation of new memories. So there was a lot of discussion and prediction that rapamycin treatment would cause dementia and impair cognitive function. Again, what we see experimentally, again, at least in rodents, is exactly the opposite, that you preserve cognition, prevent dementia in the context of aging with rapamycin treatment. So, dose, duration, tissue specificity, all of that comes into play. But again, I think it's just really important to recognize that just because mTOR is required for a certain process, partial inhibition or transient inhibition isn't necessarily going to impair that process.
Yes, I think part of the confusion is that the discussion of mTOR inhibition is often coupled with a discussion of reduction in protein. And then when you talk to somebody doing muscle research, in the case of sarcopenic folks, it's pretty clear that a relatively high protein diet is necessary or they will lose muscle. Right. And this is true across the board. So I think there's a confusion there. In other words, confusing the effects of rapamycin with the effects of not eating enough protein.
Yeah. I also think it has to do with transient versus continued inhibition of mTOR.
Yes.
So, most of the studies of rapamycin in the context of aging now, particularly in humans and also our studies in dogs, have moved to a once weekly dosing strategy with the idea that you strongly inhibit mTOR complex 1 right after dosing, but then the rapamycin levels reach zero before the next time you dose, which allows a restoration of full mTOR function. So, it's this question of hyperactivation of mTOR is pathological. We know that.
Yeah.
And too little mTOR is pathological. We know that. So, where do you strike that right balance, I think, is the question. And during normal aging, it seems to be the case that there's a drift towards hyperactivation of mTOR, at least to a certain point, but then once a pathology sets in, like sarcopenia, specifically in the muscle, you probably have too little mTOR activity in the muscle in the sarcopenic state.
And this pulsatile activation of mTOR seems interesting in it, or pulsatile deactivation, as the case may be with rapamycin. Right. And that's what you see people that are exploring rapamycin are doing. And in the opposite case, you look at bodybuilders, many of whom have mTOR essentially always activated that pathway.
Right.
Through
Yeah, that's
diet
right.
and other means, pharma, diet, other means.
Yeah, this raises a question I'm really interested in, and I have no data at this point, but it'll be interesting to see how this plays out. Because, as you just mentioned, there are hundreds, maybe thousands of people self-experimenting with rapamycin now. And the question of how does rapamycin interact with exercise, particularly resistance training, I think is a really interesting question. And of course, it's going to be dependent on dose and the pulsatile versus continuous. I sort of speculate that once weekly rapamycin, if anything, will probably be beneficial in the context of resistance training, at least in older people. And I think it's more about the effects on chronic inflammation than it is about the effects on muscle synthesis. But there's really no data, so I'm really very interested to see how that plays out.
That's a topic that folks here are interested in as well. People that do sort of molecular study of muscle.
Yeah.
Because it's really a question that's in a lot of people's minds, just the way you described it.
Yeah. Yeah. And with aging, we know that there's an infiltration of fat into muscle and probably an infiltration of activated immune cells. And I think rapamycin is going to be pretty beneficial at counteracting that.
And the
inflammation. So what the net effect? Yeah, exactly. Well, and those drive inflammation, right? I think what the net effect is, it's going to be really interesting to see.
So, Matt, we just discussed that there are hundreds, and actually I think it's thousands of people currently self-experimenting using rapamycin. And as we discussed, they typically use this sort of intermittent dosing protocol, a very modest sort of dosing. I think I know the answer to this question, but I do need to ask you, do you think we're at the point where folks like this should start seriously considering using rapamycin, or do we need a bunch more research first? There's a value in having a large community of self-experimenters, but there's a risk associated with that. And I wondered if you talk about the risk-reward trade-off there.
Sure. So, I think the first thing to say is that, I am very hesitant to make recommendations on what other people should do.
Of course.
I just don't feel like that's my place, right? I'm not an MD. So what I will say is, my own personal experience. So I take rapamycin. I do it about 10 weeks on, a few months off in this once weekly dosing. My personal experience has been that I have noticed some significant benefits, and personally, I have not experienced any side effects. But there is a risk for sure associated with taking rapamycin. I think it's small in these once weekly dosing, but I also think we have to be honest. We don't really know what the actual risk of short or especially long-term rapamycin dosing with this sort of strategy is. But the other thing I think that's important to recognize is there's a risk associated with doing nothing as you get older. And we all know where that is.
Yeah.
So when you evaluate risk reward, my personal risk reward analysis places the reward pretty high for rapamycin and the risk pretty low. We're actually trying to figure this out now. So we have a project that was funded by the Impetus Grants program that's collecting survey data from people who have been self-experimenting with rapamycin, from months to years. And so, obviously that's an imperfect kind of study, right? Of course, we want the double-blind, placebo-controlled clinical trials. But I think that, like you were suggesting, there is value in collecting information from people who have been self-experimenting with rapamycin and likely other things to try to get a feel for, how many of those people have experienced what we might consider side effects attributable to rapamycin. How many of them think they have experienced benefits? How many people stopped taking it because, they had a bad experience? So we're going to try to get that information. And again, recognizing it's going to be imperfect, but to start to get a feel for, safety, particularly whether or not there's any real risk of significant adverse events. And I think that's the thing we really want to know. Is there an uptick in risk of life-threatening sorts of things?
Right.
My intuition and everything I've learned from talking to people is the answer is probably no. But until you really try to dig into it, you aren't going to know for sure.
Yeah, you hear instances. Of course, there are individual people with individual situations where they feel they are actually perceiving a degree of benefit. Have you perceived benefit?
Yeah. So I mean, again, I'm a scientist. I certainly recognize the placebo effect is real.
Oh, huge.
But in my personal experience, the first time I started taking rapamycin, it was because I was experiencing something called frozen shoulder or adhesive capsulitis. This was a few years ago now. This is inflammation of the shoulder capsule. And it took me a long time to get it diagnosed, but I had really, really severe pain in my shoulder, and I couldn't lift my arm above my head, and I couldn't sleep because of the pain. And I finally got it diagnosed, and the specialist, was basically told me, there's not a lot that I can do for you. Sometimes it goes away in a year or two. And I was really disappointed with that diagnosis. I actually, believe it or not, was hoping I had a rotator cuff tear because I could get surgery and get it fixed. So after I got that diagnosis, I started, thinking to myself, this is a chronic age-related inflammatory condition. Rapamycin might help. And so I started taking rapamycin. And within a few weeks, I noticed a pretty significant reduction in pain. And by the end of 10 weeks, it was gone. I mean, basically, it had been completely cured and it hasn't come back. And so again, sure, that could be placebo effect, but it was so painful that I just have a hard time believing that wasn't something real from rapamycin. And biologically, it makes sense.
Yeah,
that's exactly. It's exactly the kind of indication where you might expect that rapamycin could have a positive impact.
Absolutely.
And the other thing I'll say is, I mean, again, this could be more placebo effect than reality, but I definitely perceive a decrease in joint pain. So I do a fair amount of resistance training, and when I'm not taking rapamycin, I tend to notice an increase in wrist, elbow, ankle pain when I'm lifting weights. And every time I take rapamycin, it seems to go away. So that could be placebo effect, but I kind of think it probably is just a general decrease in inflammation.
So, Matt, this article we've been talking about is fascinating, and I encourage our listeners to check it out. We'll have a link to it in our show notes. But here's the million-dollar question for you. For someone who lives by the motto, live long or die trying, what are the strategies that you use in terms of diet and supplements and exercise?
Yeah. So I actually, for this field, I guess I just admitted I take rapamycin, so maybe that makes me a little edgy, but in this field, probably not. So I don't do a lot, honestly. I try to eat a healthy diet. I found for me personally that a low carb diet tends to work pretty well. And that's mostly just trial and error throughout my life. But I've sort of found that for me works really well. I don't try to do a ketogenic diet. I do regular resistance training. So I'm pretty religious about lifting weights, 3 times a week at least. And I try to get other exercise. My wife takes me hiking, which is great. Gets me outdoors. The only supplement I take really is I take vitamin D when I remember. And again, that's because my wife tells me I should, and I live in Seattle, so I probably should. And I take rapamycin periodically. That's about it. So I don't honestly do a whole lot in terms of other anti-aging interventions that are popular, in part because, again, I'm pretty skeptical of a lot of the stuff that's out there. And I don't necessarily think it's going to hurt you, but I don't see much evidence for benefits from a lot of the other, quote unquote, anti-aging supplements that people take. Now that may change. Look, I'm 51. So as I get older, I'm sure I will become more aggressive in my attempts to not die trying. But for now, that's pretty much what I do. And I'm optimistic that the science will continue to progress and we'll have, real validated interventions and also ways to measure efficacy of these interventions going forward. We didn't talk at all about the aging clocks, but I think that's a very sort of promising area of the field to start to get a feel for what lifestyle or supplement or pharmaceutical interventions, might actually be working, especially at an individual level. I think we're really not there yet at having any sort of personalized approaches to optimal longevity or health span.
That was great. And the aging clocks are indeed fascinating. And there's a variety of them now with different characteristics.
Yeah. And I think, in some ways, the sort of popular discussion of the aging clocks has gotten way ahead of the science. But again, I think there's reason to be optimistic that the science will catch up and that we'll really have some tools. They're not going to be perfect, but we'll really have some tools to give an idea for whether or not different interventions actually work, which aspects of the biology of aging they're working on, and hopefully some sort of personalized way to tell you what an optimal approach is to your own health span.
Yeah, it'd be valuable, particularly for those that are still relatively young, At some point, I don't need a clock, right? Squat, can you squat? Can you move normally?
Yeah. Right. I mean, but also as you get older, it's easier to tell whether an intervention is working for you or not, right?
Exactly.
You maybe don't need the epigenetic clock.
That's what I mean. When you're older, it's a different ball game.
As Ken is saying this, he's looking at me, by the way.
No.
He's like, your time is coming too.
Yeah, I'm the old person here in this conversation.
Well, Matt, this has been a lot of fun. So we have just one more question for you. How's your dog doing?
Dobby's doing really good. So he's 11 and, getting older, definitely showing some of the signs of old age. But, he's doing good, getting around. It's funny because he was diagnosed with oral melanoma a few years ago, and we got past that. And then we really started to see some what looked like pretty significant declines in function. He was going downhill fast, and he's bounced back strong. And so, we're just enjoying every moment that we have with him. Of course, it's not going to be long enough, but he's doing good, and I hope we get at least a few more years with him.
That'd be great. The loss of a dog or other pet can really be painful.
Yeah, it's another family member.
Yeah. Absolutely. And I mean, that's part of what makes the Dog Aging Project so special to me, is that I recognize that, many people consider their companion animal to be part of their family. And so, regardless of what we learn about human aging, and I think we will learn a lot about human aging, if we can have an impact on the quality and quantity of life for a family member, that's important to people. And so I think that's, and I think we have a real opportunity to do that with what we're going to learn. So I'm pretty excited about that.
And that may in turn extend human health span, at least. The dog
That's
is
right.
good for us, and I think it's the only other species where you get a cross species generation of oxytocin. When
right.
you hug your dog, you benefit and the dog benefits.
Yeah, that's right. It's really interesting, the biology that happens in both the dog and the person when they interact, and it's fascinating and
it's remarkable.
I agree completely with you. Yeah, I'm going to go hug my dog as soon as we're
on
off.
that note.
Yeah, co-evolution.
That's right.
Well, Matt, it's been fantastic having you on the podcast. Thank you so much for joining us.
Sure, thank you. It's been fun.
Yeah, you were terrific, Matt.
STEM Talk. STEM Talk.
STEM Talk.
STEM Talk.
Well, Matt really does wear a lot of hats at Washington, and the range of his research is so impressive. And it's interesting how he was able to study yeast and C. elegans and eventually determine that they were key aspects of aging that are shared across all animals, all the way down to yeast. And I might have to check into his dog aging project to see if I can glean some things to help my cats live longer as well.
Well, Don, maybe you should investigate creating the cat aging project.
There you go.
There seems to be an opening there. But in all seriousness, aging research is on the cusp of a golden age. With new technology, we are accelerating our understanding of the biology of aging. What impresses me about Matt's research is that he not only focuses on understanding the nature of aging, but he is also investigating how to extend the number of healthy, active years that people, as well as dogs, can enjoy. The goal for all of us should be to improve not just lifespan, but more importantly, to improve our health span so we can enjoy a high quality of life as long as possible.
So if you enjoyed this interview as much as Ken and I did, we invite you to visit the STEM Talk webpage where you can find the show notes for this and other episodes at stemtalk.us. This is Don Cornegas signing off for now.
And this is Ken Ford saying goodbye until we meet again on STEM Talk.
Thank you for listening to STEM Talk. We want this podcast to be discovered by others, so please take a minute to go to iTunes to rate the podcast and perhaps even write a review. More information about this and other episodes can be found at our website, stemtalk.us. There, you can also find more information about the guests we interview.